Okay, now the meat and potatoes of the talk, the bioinformatics. So now we get to the point. Everything's good. You know, our data looks good. We've controlled for variables. We're seeing a statistical difference. Now what? And I think I like this quote, this lyric from Jimi Hendrix. I think this sums up basically what I do. Well, I stand up next to a mountain and I chop it down with the edge of my hand. And that's kind of what you do. You end up with these huge mountains of data, right? You know, a typical experiment, you're going to get 1,000 different significant genes. What do you do with that? You can't chase all those genes, right? We need a way to take that mountain and chop it down into something we can use, something we can publish, something we can search for as a biomarker, okay? And hopefully what I'm going to do is maybe give you some hints. And they're not necessarily this is kind of a part of what Sue and Katrina are talking about. This is my philosophy, and it might not necessarily be the standard way to do it. But again, ends justify the means. Whatever gets you to that end point is fine. So this is just kind of an overview. If we take the previous example we used, 10 tumors versus 10 controls. The first thing I do always, if possible, is eliminate the absent genes. You know, there's a lot of statisticians that'll argue you can't do that, you know, you're biasing the data, blah, blah, blah. I think, and I've seen this, is when you eliminate the absent genes, remember that beta curve, it seems to get steeper. So there's a lot of, I think, false positives that are generated looking at data that's just barely expressed or not expressed at all. So I get rid of that. And I think it cleans up a lot of my further analysis. And then from there, obviously now we're looking at the present genes. You can do a variety of statistical tests. You've got parametric, non-parametric, correlative, clustering, et cetera, et cetera, et cetera. And this can be mind-boggling. Which statistical test do I do? But whatever statistical test, your main goal is to generate a list of genes of interest. These are genes that you believe are involved in whatever particular phenotype or condition that you're studying, right? And from there, we can use bioinformatics to chip away at this, to get at, you know, what do we really want to look at? What is important in our system? And I'm going to tell you that I think you can use this to actually kind of justify some of the statistical methods that you use. And I'm going to demonstrate this here in a little bit. But first, this is my disclaimer that these views are all mine and do not necessarily reflect the views of management. That this is all based on my 10 years of doing array studies and being a bench scientist. And a lot of my views are based on the biology, okay? And the way I view the genome is kind of like a spider web, right? That, you know, this kind of tongue-in-cheek koan that I made up, you know, is kind of true, that you don't see one gene expressed, right? You know, genes impact other genes, kind of like a spider web. I pull on that spider web, we're going to get, it's going to affect the, you know, the entire web, and it's going to affect it in different ways depending on how close it is to that, where you pull it, right? That in order to perform, in order to get a phenotype, you have to have a collection of genes, and many times those genes are involved in similar pathways, or they are connected in performing a complex biological function. That there's a connectivity to these genes. And that when I compare A versus B, and I get a list of genes, these genes should be important into the system that I'm studying. And they should be connected in somewhat, more than you would expect. That gene A in my list might induce gene B, which inhibits gene C, et cetera, et cetera. So what I mean, let me give you a visual. I'm kind of murdering that. Is that if you think you've got a list of genes that are important in, say, a disease you're studying, you know, they're going to, a high proportion of them are probably going to be involved in similar biological pathways. And you can actually see that when you actually look at the array data and superimpose that on these pathways. One of the cool things, and this has kind of been a more recent development, is you can generate these networks. So here we're grouping genes associated with similar biological functions. Here we're just associate them if any biological connection exists. And typically these networks have a limited amount of genes that they'll connect. They're also called interactomes. But any kind of biological connection is drawn here. And so you can see that. And so that when I generate my list, I want to see a lot of these because I think these genes are connected and they are going together to perform a particular function, okay? Or you can actually look at just single genes. And again, this is all making footprints here, right? So here's ERB2, and here are all the genes known to interact with it, right? So the expression of ERB2 itself goes down, and you can see, green is down, red is up in expression. And you can see that the expression of this is down, but you can see a lot of its interacting partners are also down. It's leaving a footprint in your data. And you can see this. If you spend enough time, you can see these footprints. Now this is what's really cool, is not only can you see a footprint left at the scene of the crime, but you can see it from a distance, right? So here we have TGF-beta 1. And this gene is regulated post-transcriptionally. So changes in the activity of this aren't at the expression level, it's at the protein level. So we don't see this in our list, right? But if we look at all the genes known to interact with it, they all go up, right? So what would you expect? What would you say TGF-beta is doing in our system? Come on. Upregulated, increased activity. Even though in my list I'm not seeing a change in that expression, I see its footprint, right? I don't see the stone, but I can use the waves to get back to that stone. Does that make sense? And that happens a lot. I mean, if you think of it, you're studying a particular tissue, there's endocrine effects, there's growth factors coming in, blah, blah, blah. And that might be a tissue that's completely distal to what you're looking at. But we can see those effects if we actually pay attention to this footprint. And that's that connectivity, right? We've got a central molecule here connecting all this gene expression. I mean, this is what you want, right? You want to get biological context out of your genes that are different. So here's some pathways that are associated with a list of genes that are different in gonadotrope tumors, right? And there's obviously a lot of pathways that come out significant. That it's you as the experts in your field decide what I want to study, right? There's a lot of these pathways are probably different because, normal pituitary is just different physiologically than, you've got 6 cell types in a normal pituitary versus 1 cell type in a cancer, right? And so there's going to be effects of differences in cell type. But as expert in your field, you're going to know which one of these pathways you should investigate more. In particular, obviously you see this P53 signaling pathway. That might be a good, there's a history of P53 involvement in cancer. This might be something we want to investigate. But again, it's this connection. And you're getting more depth out of the list of genes you think are different in your study. So let me give you an example of that and how you can use this connectivity to help guide your analysis. Okay, remember I mentioned the 2 primary cell lines and the and the immortalized cell line stimulated with cytokines, right? What statistical test would you use to generate your list of genes that are different? Anyone? Well, remember, okay, let's go just parametric test, okay? What would be the test? T-test, but what kind of T-test? You would think, right?
Well, which way are you comparing between stimulated and controlled?
Right, these are the same samples, these are the same cell lines split in half. One was treated with a cytokine, one wasn't. So these are actually paired results, right? So I would say that from a statistical standpoint, you want to do the paired test, okay? That's what makes more sense. That to ignore that these are the same cell types, you don't want to do that in statistics. You want to account for that, right?
So you're doing the paired on the 2, so when you do them, you're doing 2.
Right, instead of saying, so in an unpaired, I would say, is this mean different than this mean? In in the paired, I'm saying, is this distance different, right? So overall means versus distance or difference in the two conditions. So I did both. I actually did both. So on the left side, I've got, so here's my paired t-test, and at a p-value of less than 0.001, I get 316 transcripts, okay? And here are the signaling pathways that I found significant in there, and here are the networks along with their scores. The higher the score, the more significant that network is. And the molecules from my list, or my genes from my list, that are associated with these particular networks, right? So I did the unpaired t-test, right? And to make it comparable comparison, I only took the first 316 transcripts. So we're dealing with the same amount of genes. Now look at this. Look at these pathways. Here's the log p-value score. The higher the score, the better, right? The more significant. You know, my top score here is 3.8. Here I've got 9.13, right? I'm getting, you know, we're getting pathways, but these seem to be more significant. And you can actually look at, so if we look at interferon signaling right here, it's got a log p-value of 3, and I've got 4 molecules. So that means 4 genes in this 316 are associated with interferon signaling, right? I look over here, where is it? There it is. I get 7, right? We go down to networks. You can see that my network scores are all bigger here. I'm showing more connectivity, biological connectivity in my data, using the unpaired versus the paired, right? And I'm also getting more of these molecules that are actually in these networks. So based on this, which statistical test would you rather use? Paired, or unpaired, I mean, right? Sorry, now you got me going. So here's an example. So this was the interferon signaling. I got this switch. This should be paired and this should be unpaired. But in this particular list that we've drawn, we've got all these genes that are coming up. If it's colored, it was on our list of genes that were different. And so we're losing this one in this diagram, but we're gaining these receptors here. We're also gaining some of these downstream effects. And in my mind, the more pieces of the puzzle you have, the better the picture. That's what I'm trying to get at. So, you know, this was connectivity of different genes. What about the gene itself or the product that gene makes? We can use that to kind of narrow down our list. And typically when you'll put a list of genes into your bioinformatics software, you get something like this. You'll get the gene symbol, gene name, fold change, its associated p-value. But you also get this, and most people just ignore this. You'll get its cellular location. There'll be a column here showing its cellular location. You can get types. Is it a transcription regulator, a kinase? And this is new, and this is on Ingenuity. I don't know if it's going to be on the others. I'm sure it will. But has this gene been used in a biomarker study? So here, location, cytoplasm, extracellular space, nucleus, plasma membrane. You know, if I'm looking for a target for a chemotherapy agent on cancer, I might want to look at, you know, these genes on the plasma membrane might be particularly interesting, right? And say we know it's a G protein receptor, but we don't know what it is. We can look at this too. You know, you can just look at all your kinases, ion channels, you know, enzymes, cytokines. If you're studying some kind of infection, obviously the cytokine group might be interesting. You might want to look at that just by itself. Again, this is just ways of chopping down the mountain. And again, if I have and this actually happened, is we're trying to find markers of aggressiveness of tumors. And we had a list of like 64 genes that we thought, you know, were different or would distinguish aggressive versus non-aggressive tumors. And a lot of those genes were also diagnostic for other cancers. And that's a good sign. You know, you're using biological information to not only make yourself feel good about the list that you're generating, but I mean, it guides the analysis. Another way to segregate, and I don't think a lot of people do, is using an all or nothing type of comparison, right? That if I'm studying a tumor and a control, and I have, in my tumor, I have this gene's being expressed, but in the normal, it's not being expressed at all, I think that's pretty significant. When the light's on and off versus, is it dim this or that, can play a huge role. Like if I'm going for a biomarker, this is the perfect biomarker, right? It's either there or it's not. And you can use this, and we've actually used this, that in our tumors that we studied, we found this growth factor, neuregulin-1, which isn't in the control tumors at all. And it's actually proven to be a very important in the tumor genesis of this particular tumor we're studying. So what if you don't have, say, something to compare to? And this happens occasionally, right? So here I'm given 28 lung cancer cell lines. I don't have any normals in here. What to do, right? Well, I guess we could look at the different subtypes and do our comparisons there. We do hierarchical clustering. But we can also segregate by coefficient of variance. Does everybody know what coefficient of variance is? It's basically your standard deviation divided by your mean, right? So it's kind of your variance across the mean, right? So I want to look at things. So we know that these lung cancer tumors vary widely, that different therapeutic or chemo agents work on different types of tumors, right? So let's explore that difference. And so we can actually, I say, I can put my criteria. I only want to look at those transcripts that vary, have a CV greater than 20%. And then we can put that in our bioinformatics. And we'll get these pathways here that come out significant. And this is a hepatic cell activation. And this comes up a lot when you're looking at cancer cell lines, because it has this pathway has a lot of growth things. And you can see in this lung cancer cell line, I know this is hard to see, maybe you can see it on your printout, but these are all different growth pathways, signaling growth pathways. That's cool. This is meaning that these cell lines are probably using different ways to grow, or signaling to grow, right? And I don't show this here, but there's actually a correlation. Like if TGF-beta's up, interleukin-4 will be down. And you can actually kind of flavor your cell lines that way by biological or by pathways.